Looks like we've got a number of people here already. So I think we can probably go ahead and get started. So good morning, good afternoon, everyone. As usual, I'll just kind of go through a bit of a summary of what's gone on over the last couple of months, and then I'll turn it over to Frank and the UHN team to go through the synthesis of some analogs. So we do have some analogs that have been submitted, and Rachel and Feng Ling will kind of go through those assay results of the new compounds that have been submitted. And then, essentially we'll just kind of finish off with next steps. Variational team isn't here today, but they are kind of finalizing a collaboration with Enemy. So I'll give a brief update on that. And then I think we'll need to, discuss future steps in modeling and synthesis, and finally data sharing. So we do have a CTD vault that now has a population of compounds, and that resides at UHN, and we'll need to start uploading some of the biochemical data and eventually start sharing that on a platform such as Zenodo. So in terms of a summary, as I mentioned, we have the UHN group has submitted, I guess, 13 analogs that have now been tested. And I would say an early read on the SAR shows that small substitutions in that C ring, basically replacing the bromide on the thiophene ring, are tolerated. So we do have some results, and we'll discuss which compounds we can progress into selectivity assays and potentially into cell-based assays as well. In terms of the second round of modeling, we discussed as at the last meeting, and I ordered a number of starting materials. And unfortunately, we've had some issues with getting those across the border. So essentially what's happened is I've ordered compounds through Agora. We don't have an agreement with a customs broker, so generally the shippers such as FedEx will act as a broker on my behalf. I guess due to the new world order right now, that FedEx no longer will act as a broker as of June. So unfortunately, my shipment got kind of caught up in this. UHN has agreed to use their broker to release the shipment. And as of this week, it looks like things are progressing, but it's still at the border and it's been there for close to 2 months now. So hopefully that shipment comes in, and the chemists will have a new batch of compounds to work on. But moving forward, I think Frank and I have discussed probably the best strategy will be to order compounds through UHN, use UHN's established broker, and any costs associated with those chemicals we can reimburse from the Agora account. So in terms of modeling, Variational has started some refinement on the PRMT6 model using Mo. So they kind of created hundreds of different configurations to look at the missing loop with both de novo design and from the PDB. And following a visual inspection, did some more interactive minimizations with their software, and these results were then shared with Conrad. So in the next steps, I guess, Frank, would you want me to drive, or do you want to start sharing the screen? Or who from the UHN team will actually present this work today? I think we have a couple of new people joining us, Connor O'Shea.
Yeah, you can continue the presentation, and I'll just go next slide or something.
Sure.
Chemistry should be pretty quick. So I showed this last time during our meeting. This is kind of the general synthesis that we've been using to generate some of these analogs for PRMT6. So In our last meeting, we kind of discussed a little bit of a scale-up issue in terms of some of the steps. In terms of yields, we have since kind of remedied that. So you'll see the first green yard step here. On scale, we've gotten it up to about 50%. And then the next step, which is the alkylation and ring closing step to form the benzodiazepine, on scale now is roughly around 60%, which is much better than previous 27% that we've been getting. So with that reduction step of the imine to the amine proceeds quite well, and then we simply do amine coupling with various acids that Pete has been acquiring for us. So if you go to the next slide, these are the compounds that we submitted a few weeks ago, early September, to Rachel's group, and along with a few more compounds that were resubmitted in July, and I'll let their group present some of the data associated with each of these compounds. And yeah, so in terms of the first group of acids, we're almost done making the first set of acids. Obviously, with the shipment delay, we'll wait and see whenever those arrive, we can continue the synthesis of that. We are in the background trying to come up with different ways to potentially synthesize the benzodiazepine core, like Pete was pointing out there, by incorporating that into stereocenter in the synthesis. So right now we're just making racemic mixtures, and so in the background we're trying to optimize some routes potentially to get a better way at getting into that core.
Frank, quick question: what's the BBB score on these, or CNS NPO, just to get an idea of potential CNS penetration? We've calculated it.
No, but if in the next slide, we did do a short PK on the SGC compound, just to give us an idea of exposure. And so we'll see at 5 mg per kg via IP route. This is the SGC compound. Overall exposure is quite low in both the brain and the plasma. So I think that's kind of our answer there. So we can try to optimize that as well.
Yeah.
Well, yeah, I mean, it gives you a benchmark. I'm just wondering generally how these score. Just trying to get an idea on a scale of 1 to 6. What they calculate as anyway.
Yeah, I haven't calculated, but I can do that.
Yeah.
But I guess even though we do see some low exposure, the brain to plasma ratio is, we are seeing exposure in the CNS, so it's at least a good sign early on.
Yeah, we should probably correct that for free drug though, just in case. Mm-hmm. Not just in case, I mean, we should anyway, but it might change the complexion of that once you get brain binding and plasma binding. Yeah.
Okay, that's all for my side.
So have you started any of the, I guess, asymmetric synthesis or looking at ways to make that, or is that all just kind of in the planning stage right now?
No, it's ongoing. So I think we've tried a few things. I don't think anything has stuck just yet, but they're ongoing right now.
So you're looking at that by chiral HPLC? Or
right now we're looking at asymmetric reduction of that imine. We're also potentially looking at forming that cyclic benzodiazepine in an anti-selective fashion as well. So there's a couple of options.
Yeah, but at this point you don't have I guess a method to see how you're what your enantiomer selectivity or enantiomeric excess is going to be at this point. So you would need a chiral HPLC method or some other method to look at that.
Yeah, so I know we have a chiral column that we can use, so we can start developing that method as well.
Okay. Okay. Any questions for the chemistry team? Fengling, do you want me to stop sharing and have you share your slides, or I do have the slides that you had sent, but
sure. I just want to share the updated one.
Sure.
You see?
Yep.
How to make the full.
We see it full screen on our end, Fengling.
Okay.
Yep.
That's great.
Thank you.
So, my name is Fengling. I'm a research associate at SGC, working on the in vitro assay development and the small molecule library screening using biochemical or biophysical methods. So for this PMT6 project, before presentation the assay data, I want to introduction the general background information for PMT6 and its assay. Next. So PMT6 is a type 1 arginine methyltransferase. It modifies both histone and no-histone proteins by the methylation. It's a function involved in various cancers and regulation different genes. So PMT6 is a potential drug target for cancer therapy and the antiviral treatment. About the PMT6 catalyzed reaction. In this reaction, SAM was used as a cofactor. SAM is a methyl group donor. This methyl group labeled with a tritium. And the built-in labeled peptide or protein use the substrate. After PMT6 catalyzes this reaction, the product methylated peptide can be measured by detection radioactivity signal. In the detection, we used SPA technology. In this detection system, we just use the SPA beads. Only methylated peptide is binding to the surface of the beads. So it makes the radioactive, the methyl group, close enough to the surface of the beads. That generate radioactivity signal. The free hot SAM and the no methylated peptide cannot generate any signal. So we just measure the radioactivity signal to check PMT6 activity. So we use this assay condition, tested both substrates, histone H3 and H4 peptides. From this data, peptide H4 showing more active than peptide H3. So we use the H4 peptide as a substrate, developed PMT6 in vitro assay conditions. Using the optimized assay conditions, we developed 3 PMT6 probes. MS023, MS049, and SGC6070. So the probe SGC6870, we used this probe as a parent scaffold for the design and the synthesis of new analogs, just like Frank just presentation that the chemistry. So here the data is showing that the IC50, the values using the different pre-incubation times. You see, without pre-incubation, this compound not very potent. The IC50 7.4 micromolar. But after 2 hours pre-incubation, IC50 is a 60 nanomolar. So it's a more than 100 times different potency. So this probe is a time-dependent inhibition on PMT6. But this binding to PMT6 is not covalent binding. We already confirmed by the mass spec. So because this inhibition is time dependent, so next we tested these 2 batch compounds using the 1 hour pre-incubation. So these 13 compounds inhibition data, the top 9 compound is showing a very similar potency from 0.3 to 1.5 micromolar IC50. This control is a probe. This inhibition dose response curve against the PMT6, bench 1 compounds, and the bench 2 compounds. Yeah, that's all data. Any questions?
Peter, I think you're muted.
Thanks for that. I should know better. I just had one question about the hill slopes.
Hill slope.
Yes, I mean some of them are quite, I guess, gradual. Is that because you see that it's time-dependent inhibition?
Yeah, because from this hill slope, the almost is good because the hill slope from 0.5 to 1.5 to 1.5 is good, but this one is a 2.6. That's because the compound concentration range use not good. If I use a smaller concentration range, maybe that the hill slope improved.
Okay.
You see this control. I just use the hill slope is good because I use the highest concentration 5 micromolar. But this one, the highest concentration 50.
Okay. Any additional questions for the biochemical team? I'm going to try and...
Thank you.
Thank you, Fengling. That was super.
Yeah. I'm trying to share my screen again, but I'm having some issues here. Sorry about this. I'm unable to share my screen right now. Okay. Back in business. So I just kind of summarized that data that Fengling had presented, and we had a kind of an arbitrary cutoff of 1 micromolar to kind of move forward into the next step for these racemates. So, you know, as Frank mentioned, these compounds are isolated as racemates, not asymmetrically pure compounds. So, you know, at moving forward, if we were to separate out the active enantiomers, obviously these numbers are going to come down somewhat. So essentially what I've kind of summarized here is if you look at where we've replaced this bromine with an alkyl group, moving the sulfur, you know, around the ring, I would say we do see at least a retention in activity compared to the probe compound. You might make the argument that when you actually remove some of that that bromine, then you are seeing a decrease in potency. When we move to a different chemotype, again, they're kind of in the same range, but you are starting to see maybe a little bit of drop off in potency. But I think what we can say from at least these last set of compounds is if you had something a little bulkier and maybe a little bit more greasy, potentially if you have a hydrogen bond donor in this region, and certainly if you increase the bulk here, we are seeing a dramatic drop in potency. So I think moving forward into the selectivity assay, I would suggest we, at a minimum, move ahead with these top 4 compounds. I think we could probably make the argument that, you know, all these, though these are a little weak, there is a bit of a different chemotype here in some of these rings. So I'd be supportive of moving those forward, but if, you know, if there are some compounds that come in the next group and we decide we only want to have a plate of maybe 8 to 10, certainly we could discuss dropping some of those compounds. And just a reminder, this is kind of the flow progression that we had for these compounds. So, you know, this is a little bit arbitrary. I put 1 micromolar for these compounds that are racemates. If they were pure compounds, pure enantiomerically pure compounds, we certainly have a target product profile where this would be less. But I think for this early stage of the program, then 1 micromolar cutoff is reasonable. So maybe I'll just turn it over to Rachel's group. Is it 10 compounds for selectivity? Is that a reasonable number?
I mean, Fengling can comment better as she's the person doing the experiments, but I think 10 compounds is a reasonable throughput. Yeah.
Yeah.
Yes, yes.
Okay. And Frank, we have a few other compounds that are coming down the pipe that, you know, maybe might bump a couple of these ones.
Yeah, we should have a few more close to being done.
Okay. So yeah, we'll make that an action to move move these compounds into the next set of assays. So that's basically the update on new work that's been done in the past couple of months, and then moving forward, as I mentioned, the variational group is actually meeting with Enamine at a conference in Boston right now. So there is, you know, in principle, an agreement to run a collaboration with Variational using their modeling platform and Enamine's build design space. So essentially looking at their library of commercially available compounds and analogs that are kind of straightforward to synthesize as the first iteration of modeling. So hopefully that's you know, going to be coming together quite quickly. For compound synthesis, we kind of touched on we do have plans, as provided those starting materials get here soon. But I think in terms of next steps for modeling, I mean, it is only a few compounds. We can look back at what's been done already minimally for the generation of this probe, but it does at least appear in this region there isn't a great deal of space. But we certainly could consider a modeling exercise where we move this amide and then build a library based on just our groups attached directly to that nitrogen. Those compounds could feasibly be made from this starting material, doing reductive amination with a ketone or an aldehyde. And there are some papers that people have published with that chemistry on a very similar core. So I think that would be quite easy to do. So we can certainly send a list to Conrad for modeling on something like that if people are in agreement. And really the last thing that I wanted to talk about is data sharing. So we do have the CDD vault at UHN. Currently, it's just populated with the compounds at this point. I guess kind of want to open it up to discussion on how or who would be responsible for uploading the biochemical data. I know that we discussed early on giving access to someone from the SGC to have access to the CDD vault like you've given to myself. So if that's something that can be done either at the SGC or if Frank's group can take the data and upload it, one of those two scenarios I think would work.
I mean, I can comment. Yeah, I mean, so we have access, Frank, right? So we're using effectively one seat for this, and then Pete, you have one seat. Is that right?
That's correct. Yeah.
Yeah. So someone at SGC is actually going to be uploading the IC50 data. It's probably good that they have access. So Frank can put in the data, put in the molecules, create the the fields, but it would be probably better if someone SGC actually uploaded the biochemical data, and we can sort out a training session if needed how to do that. Like I think CDD Vault would help with that. But that's probably the most proficient way to do it. I think we could probably add the DMPK data as it comes in, right, Frank? Mm-hmm. So we can add the structures, the DMPK, but it's probably better for someone who knows how to handle the data to upload the biochemistry.
Yeah, I would suggest Fengling could do that, but I'll discuss with Fengling and then we'll decide who is best positioned to do it. Yeah, because I think she's already uploading the data anyway into our SGC database. So
yes,
yeah, so everything's already she has everything formatted and ready to go.
Cool. Yeah, so Frank, you could probably send an invite to Fengling. And then, if you have the user ID, then I think, Rachel, you could probably get access to that and just log in. I mean, that should be fine.
Yeah, as long as we don't want to scupper your license by sharing the login, but we will probably do that. Yeah.
should be okay. Should be okay.
I'll send a login registration today.
Okay, well that was fairly quick. I don't have anything else that I wanted to bring up, but if anyone else does, certainly feel free. Otherwise, I think we can probably end the meeting and let everyone get on with their day.
Perhaps can we jump back to the selectivity panel? So, I mean, we have a huge number of methyltransferases. I think probably the most efficient way for us to screen for selectivity would be perhaps to hone in on a subgroup which are the most structurally similar to PRMT6. Some I'm not expecting to see any.
Yeah, I agree. I don't think you need to do a big panel at this point. I thought. Yeah, I think we might have discussed that a while ago, but
yeah,
I think you had maybe 5 or 6 that you were going to key in on.
Exactly. Yeah, I just wanted to double check that that's still the plan.
Yeah,
that would just also save us costs. I think
yep.
the other issue is that the tritiated SAM is, I think, about 3 times more expensive than it was before COVID. So that's really fun.
Not 3, it's a 6.5 times higher.
Wow.
Yeah, so we just have to be I'm just like mindful of resources. I just want to do the experiments that make sense without you know, it doesn't make sense to me that some of these methyltransferases are even worth looking at. I think when we get further forward in terms of potency and so forth, then we can do a full panel for sure.
Yeah.
But I think for now, like, it just doesn't make sense to me, if that's okay with you.
No, I agree 100%.
Super duper.
I have one question for you, Rachel. Sorry, because I might have missed a meeting, but it sort of came up in the discussion I was having with someone by email. Did we sort of finalize what the CNS profile needs to look like for these small molecules? Like, you know, we were talking about, do they need to enter the central nervous system or do they not need to enter? Do we have some clarity on that?
Yeah, so I mean, yes and no. So I spoke to field experts for clarity, and I received differing opinions. So some folks were very clear that the CNS penetration would be really critical. Other folks were much more convinced that muscle penetration is going to be really critical as well. So I think we don't really know, to be honest. And I think this is kind of just like the nature of working on this kind of tricky disease where there isn't that kind of information. My bet would be that CNS penetration is going to be is something that we should definitely be aiming for. Whilst, you know, we see some of the largest phenotypes arising through changes to muscle cells, I think the changes to the central nervous system, I think, are pretty profound, and that is what makes sense to me.
Yeah, it probably makes sense to have a TPP that supports that right now. And I guess if we get to a point where we've got some good robust POC models, we can put something on it or something, right, to keep it out of the CNS, or, you know, put some sort of inhibitor in to prevent it crossing the brain or something like that. We could figure that out.
Yeah. And I'm just very top of my mind this morning is, I don't know if anyone saw, there's big news for another repeat expansion disease. So Huntington's disease had their top line data from Unicure, and a big caveat, what the folks thought was, well, there are many caveats about their approach, in my opinion, but one of the caveats about their drug is it's delivered only into the striatal region of the brain, and the penetration through the different brain structures is actually, well, we suspect it's quite low based on what we know from animal studies. They don't actually have data on that for people, I don't believe, or not robust data from a larger sample at least. But they are seeing really significant effects just by targeting the most affected regions, which is really interesting. And it suggests that, you know, this precise targeting is actually probably going to be quite important. But yeah, we don't know yet. But anyway, it's quite exciting. I just wanted to share that because that's something I've been thinking about a lot since the press release. Yeah.
I saw this morning. I just shared the link. It's just, it's in the media right now, right?
Yeah.
Yeah. But again, it kind of also drives home like why I think our approach and this kind of small molecule approach is so important, because regardless of how well people like Unicure do with their gene therapies, the, you know, access and equitable, you know, the ability of people to get hold of this drug is going to be a major challenge and hurdle, even if it is a quote unquote cure. Yeah.
Yeah. No, that's great.
Yeah, that's why I'm writing my grant applications anyway for our Huntington work. So,
yeah, I mean, it's an 8-hour brain surgery. I mean, come on. It's like, you can beat that with a small molecule, surely.
Yeah.
Yeah.
One just last question for Conrad. Conrad, do you have some capacity if we send you a list of compounds for modeling?
Yes, at least I have some time now, and the resources available.
All right, so we'll work on that.
Yeah.
All right. Well, thanks everyone, and enjoy the rest of your day.
Thanks, Peter.
All right.
Bye.
Thanks. Bye. Thank you so much.